Okay, hello everyone. Welcome back to the course Fundamentals of Protein Chemistry from NPTEL, and this is week 7 question answer session in which we'll talk about enzyme kinetics and inhibition. I'm very sorry about the delay, and let's carry on with the class. Okay. All right. So in today's class, we'll talk about enzymes once again. Yes. Good evening to you. Okay. In the last class, we had talked about enzyme activity and we had solved a few questions as well, right? And in the last class, in the slides, I think I had provided you with a lot of information on enzyme kinetics and inhibition. So, funnily, it was the topic for your next week's session. And so, today we are back with those topics once again, and we'll look into them in details. Okay. So before we get started with our class today, we'll go back to last week's question. There was one question I remember in which, yes, good evening to you, good evening. So in one of the questions I remember there was a question in which they were asking that a particular enzyme needs to be complementary to the product, the transition state or the substrate. So preferably it should be complementary to the transition state and not the substrate. Okay, because there is induced pit mechanism of substrate enzyme binding. So the substrate, even more than the substrate, the complementarity should be focused on the transition state for the enzyme. So that is one tiny correction that I had to make. And now let us start with the class today. Okay. Okay. So I'll not be sharing the screen in the form of a slideshow. And let's start, because you know that there can be delay with the changing slides, and so we'll go this way. Okay. All right. So let's start talking about enzyme kinetics. Now, if you have any doubts, please stop me in the middle. You can either write down your doubts in the chat box, or you can raise your hand and you can ask me questions. Okay. If I am not able to answer in today's class, I will answer your questions in the next class. So you're always welcome to ask questions and to give me any kind of suggestions that you want to give. Okay. Okay, so the first thing about enzyme kinetics is the Michaelis-Menten equation. Now, Michaelis and Menten are two scientists who described the velocity with which a reaction proceeds with respect to the substrate concentration. Okay, so V naught is the initial velocity. Now, mind that they are only talking about initial velocity and not the final velocity or the velocity at the intermediate stage. So it is only initial velocity. Then there is maximal velocity that can be attained, and then there is Km. So Km is a constant. This is called Michaelis constant, and Km is nothing but the summation of the rates of different reactions. So we will look into that very soon. And then there is substrate concentration. So this is the graph that you are seeing, and Km actually tells you about the activity of an enzyme. So this tells you about the affinity of the enzyme for its substrate. If Km is less, that means the enzyme has very high affinity for its substrate, and very easily half of the velocity of the reaction will be attained with small amount of substrate concentration, right? And it is just the opposite for enzyme with high Km for a particular substrate. And so in that case, large amount of substrate will be required to reach half of the maximal velocity of the reaction using that particular substrate concentration. Okay, now we will look at Km in more details. So Km is equal to K minus 1 plus K2 divided by K1. These are rate constants of different reactions. We will have a look at them. So see, this is the equation where enzyme is interacting with substrate, and this is the forward reaction which is leading to the formation of the ES complex, which is your enzyme substrate complex, right? And the rate of this reaction is K1, and this enzyme substrate complex can be broken down into enzyme and substrate once again, and this rate of backward reaction is K minus 1. Similarly, enzyme and substrate can also proceed towards the enzyme and product, and that rate is given as K2. So this Km is equal to K minus 1 plus K2 divided by K1. So that means the rate of breakdown of the ES complex divided by the rate of formation of the ES complex. That is the ratio which Km tells us. Okay, right. Now there are many assumptions of the Michaelis-Menten equation. So amongst the different assumptions, the most important, there are several assumptions. Okay, the most important is a steady state hypothesis. So what is steady state hypothesis? The concentration of the enzyme substrate complex, this more or less remains constant throughout the reaction. Okay, this is called the steady state hypothesis. And initially at the start of the reaction, we assume that there is no product. So that is one. Steady state hypothesis is 2. Then the amount of substrate is supposed to be very high compared to the amount of enzyme present. And so you see that the amount of enzyme substrate complex that gets formed, that is also very less compared to the total amount of substrate that is present. Then the total enzyme in the reaction, it is either present in free state or it is present in enzyme and substrate bound state. So your total enzyme, that is E total, is equal to E plus ES. Okay, these are the different assumptions. We'll come across more assumptions very soon, and then we'll discuss about them. Okay, so until now, these are the few assumptions that are there. Okay, so during the reaction, an equilibrium condition is established for the binding and dissociation of the enzyme substrate complex. This is called the Briggs-Haldane hypothesis or Briggs-Haldane assumption, and the rate of formation of the ES complex is equal to the rate of dissociation plus breakdown. Okay, that is the steady state hypothesis. Okay, now we will look at the Michaelis-Menten constant. So until now, we have talked about what Michaelis-Menten constant is and what it tells us about a particular enzyme substrate interaction. Okay, so you know that it is K minus 1 plus K2 divided by K1, and its unit is moles, and this is the rate of breakdown of the ES complex, and the denominator tells you about the rate of the formation of the ES complex, right? Now, okay, so the characteristics of Km. So Km is the Michaelis constant, and it is characterized, it is the characteristic of the enzyme and its particular substrate, and it reflects the affinity of the enzyme for that substrate. This we know. Now Km is exactly numerically equal to the amount of substrate needed for half of the velocity to be attained, half of maximum velocity to be attained. And Km does not vary with the enzyme concentration. Okay. So, small Km tells you that very small amount of substrate is needed to get half of maximal velocity. That means there is high affinity between the enzyme and the substrate. Similarly, large Km tells you that a lot of substrate is required to attain that half Vmax. Okay. Now we will move on to some more equations. So see, Vmax is equal to K2 into E total. We know what K2 is. K2 is the rate constant for the formation of enzyme plus product from the ES complex. Okay. Now, how do we get that Km is equal to half of Vmax? So that is shown here. So when you substitute this V with half of Vmax, then you get K is equal to substrate concentration. So at half of Vmax, we are getting the substrate concentration as Km. Okay. Besides that, why Km? How Km is related to dissociation constant, that is given here. You can read about this. Okay. And now we will go to an MCQ. Okay. This is a question on enzyme kinetics and they are asking you enzyme velocity can diminish over time due to. So these are the different possibilities. Which one do you think would be correct? Okay. You can answer in the chat box. You can write down in the chat box and then we'll discuss. Okay. So the different options are decrease in the substrate concentration, product inhibition, pH changes, and thermal inactivation. So which of these different factors do you think can contribute towards decrease in the enzyme velocity over time? You can answer in the chat box.
Ma'am, option A.
Option A. So option A is 1 and 2. That is decrease in substrate concentration and 2 is product inhibition. Okay. Achha. So I am getting answers as C and A. Now let us see. Okay. Achha. So the right answer is C because all of these are involved in reducing the enzyme activity over time. Okay. If you go back to these slides from our previous lecture, then you will be able to relate. So in the last lecture, we had studied the different factors that affect enzyme activity, right? So amongst them were temperature, pH, and enzyme concentration and substrate concentration. So you know that these 4 factors are definitely involved in changing the enzyme activity or enzyme rate or velocity over time, right? And what else is there? There is also product inhibition. So we'll read about product inhibition just now. Okay. So this is a very interesting thing. See, what happens is an enzyme is equipped to get something done in our body, right? And once it has performed its function, then what happens? The product itself goes and inhibits the enzyme from doing any more of its function. This is called feedback inhibition or product inhibition. Okay. And there is one more thing that is called feedback repression. So this is an example. I am sure you all know about glycolysis pathway. So what happens in glycolysis is that we know that the product is pyruvate and ATP. Right? ATP is produced during glycolysis. It is a basically biochemical process in which sugar is broken down to give energy in our body. So what happens is this ATP goes and there is a kind of feedback inhibition and it prevents the enzyme phosphofructokinase to perform its function. So this is a kind of product inhibition seen in our body. Besides this, now this is acting at the protein level. Now what happens if the product goes and acts at the genetic level? Now the product is going and telling the gene that you do not need to express, so that less of the enzyme gets produced and less of the reaction proceeds. In our body, remember, everything is dynamic. So a particular protein exists only when it is needed. A particular gene expression happens only when that is needed by our body. Everything is controlled by our epigenetics. And so these epigenetic factors, the chromatin modifications, there can be post-translational modifications on proteins, there can be modifications on the genes, and then there can be different kinds of epigenetic modulators that prevent untimely expression of genes. Now, when this is happening at the genetic level, this is called feedback repression. So what is happening? The product is going and it is preventing gene expression. That is feedback repression. If it is preventing the enzyme from functioning, it is feedback inhibition. Okay, I hope that is clear to you. So these are the different ways in which you can regulate an enzyme's activity. Okay, besides allosteric regulation, there is also product or feedback inhibition. There is also feedback repression, in which the gene coding for that particular enzyme is completely repressed by the product. Okay, right. So we'll move on to the next question. Now this next question is again on Michaelis-Menten kinetics. Okay, and they are asking you deviation from the Michaelis-Menten kinetics occurs when 1. There are multiple substrates. 2. The substrate binding is irreversible. 3. K2 is equal to Kcat, and the enzyme displays cooperativity. Now, whatever we have studied until now from the Michaelis-Menten equation, if you see anything different from whatever we have studied, that is something that is a causative agent for deviation. Okay. Try to think. Okay. So we are getting C as the right answer. Yeah. Okay. Great. Let's see. Yes, you guys are absolutely correct. It is C. Now, why is that C? Because see, Michaelis-Menten equation does not talk about multiple substrates. It talks only about a single substrate. Okay. It does not talk about multiple substrate. It talks only about a single substrate. That is also one assumption, that there is only a single substrate involved in the reaction. If there are multiple substrates, then the entire thing becomes more complicated, and Michaelis-Menten kinetics is not able to explain that. Right? Now, substrate binding is reversible. We always know that the enzyme is binding to the substrate and later on it can even dissociate. Right? So it is always reversible. But then if it is irreversible, then Michaelis-Menten cannot explain that. K2 is equal to Kcat, that is true. That is definitely true. Okay. Now, the enzyme displays cooperativity. Michaelis-Menten equation is mostly valid for enzymes that do not show any kind of allosteric interaction or cooperativity. Okay, so single enzyme, single substrate interaction, that is very well explained by Michaelis-Menten equation. And therefore, all of these three are deviations from the Michaelis-Menten kinetics. Okay, right. So we have some more assumptions, and we already talked about this. The relative concentration of the enzyme and the substrate. You know that the substrate is much more than the enzyme. Then there is the steady state assumption, which says that the enzyme substrate complex does not change over time, and so the rate of formation is equal to the rate of dissociation, which is your K2 and K minus 1, right? Then we go to initial velocity. Always remember that Michaelis-Menten kinetics is either talking about initial velocity or maximal velocity, and also half of maximal velocity. Plus, at the start of the reaction, it is always assumed that the amount of product present is nil. Right. So now you try to work on this next question. Okay. So this is the entire equation is given for enzyme substrate interaction and product formation. So now they are telling you that the reaction shown represents the formation of an enzyme substrate complex followed by the breakdown of the enzyme substrate complex and release of the product T. The rate of the enzyme substrate breakdown will be. Okay, you're saying it is C and you're saying it's A. Okay. A. Yes, yes, good. Okay, we'll take a look at the right answer. Now we know that this is the enzyme substrate complex. Now the rate of formation is either this or this, right? But then it is an assumption of the Michaelis-Menten equation that this does not exist and the enzyme substrate is said to be converted into enzyme and product through an unidirectional reaction, and there is no backward reaction. Okay, so we kind of omit this for now. So therefore formation is only guided by K1, but then breakdown is guided by both K2 and K minus 1, breakdown of the ES complex, right? So your right answer should be A because it is a summation of both. Okay, if you look at, if you write down the formula for K1 and the entire equilibrium state, what is going to happen, then you will see that this is the right answer. Okay, now we'll move on to enzyme inhibition. Now enzyme inhibition is something that you already have studied in school, right? So this should be a quick revision for us. Okay, this should be a quick revision for us. Now I remember a lot of us used to be very confused about non-competitive and uncompetitive inhibition. So today let's clear out that doubt. Okay, so what happens in competitive inhibition? Competitive inhibition is very simple. The inhibitor is looking similar to the substrate. Inhibitor comes and bind to the substrate binding site of the enzyme. Okay, but then what happens if the inhibitor is not binding to the substrate binding site, but it is going and binding to a different site? Okay, so what happens is non-competitive inhibitor.
Ma'am, your voice is not audible. Hello, ma'am.
Yes.
Hello.
Yeah,
Ma'am, your
yeah.
voice is not audible. Can you
My
please
voice
repeat?
is not audible. Okay, okay.
Yes, ma'am. Please repeat.
Acha. Right. So, uh, what happens in competitive inhibition is that the inhibitor, it looks very similar to your substrate, and inhibitor goes and binds to the substrate binding site of the enzyme. That happens in competitive inhibition. But then what happens in non-competitive inhibition? So non-competitive inhibition is a very special kind of uncompetitive inhibition. In uncompetitive inhibition, the inhibitor goes and binds to a different region. Let's say it is going and binding here. It is binding to a different region compared to the enzyme substrate binding site. Okay, and it is only binding to the enzyme substrate complex. So inhibitor is only binding to the ES complex. If you see this figure, this is the inhibitor, the orange one is the inhibitor, and it is binding to the enzyme substrate complex and rendering the enzyme inactive. What happens in case of non-competitive inhibitor is that the inhibitor can come and bind to the free enzyme. Inhibitor can also come and bind to the enzyme substrate complex. Okay, and if it is binding to the free enzyme also, it is kind of not preventing the binding of the substrate to the free enzyme. Okay, it is not binding at the region where the substrate is supposed to bind. Now see what happens here. So this is your non-competitive inhibitor. This is enzyme, this is the substrate, and it can either be this way that the inhibitor is binding to the free enzyme, and it can also be this way that the inhibitor is binding to the enzyme substrate complex. Ultimately what happens is the inhibitor binds to the enzyme or enzyme substrate complex at an allosteric site. Catalysis is prevented due to a change in the enzyme conformation. So the product formation is not happening in both the cases. Now what happens in uncompetitive inhibitor is that Uncompetitive inhibition is that the inhibitor only binds to the enzyme substrate complex, and once again we do not get any enzyme and product formation. Yes, anything you want to say? No. Okay. Okay, we'll continue then. If you have anything to say, you can please ask me. Okay, anything, any suggestion, anything that you're not understanding, please ask me. Inhibitor binds before or after enzyme substrate complex. See, there are two different things. In uncompetitive inhibition, inhibitor binds only to the enzyme substrate complex. But then in case of non-competitive inhibition, it can bind either to free enzyme, that is before the complex formation, or it can also bind to the enzyme substrate complex. Okay. Got it? Two different things are happening in two different cases. Okay. Now this is all about competitive inhibition. What happens in competitive inhibition? See, the product is going and binding at the substrate binding site, because of which the affinity of the enzyme for the substrate is reducing. Because of that, your Km is increasing in case of a competitive inhibition. And what happens? The maximal velocity Vmax, this is also decreasing. So with competitive inhibitor, it is decreasing. So maximal velocity is the same in the, maximal velocity is same in the presence of the competitive inhibitor, but then Km is increased. Okay. This is the Lineweaver-Burke plot. Here also we see that the Vmax is the same. So Vmax is not changed in case of competitive inhibition, but then Km is definitely increased. It is becoming more positive in this case, right? Okay. Now we come to competitive, non-competitive inhibition. So in non-competitive inhibition, the Vmax is significantly decreased in the presence of a non-competitive inhibition. This is the difference that you can see. Km, of course, it is unchanged, and what happens here is, once again, you can see that there is significant decrease in the Vmax. Okay. So this is a summary slide. Sites differ in competitive and non-competitive. Yes, in competitive and non-competitive, the site of binding of the inhibitor is different. Okay, in non-competitive inhibitor, the inhibitor is going and binding to an allosteric site, but then your substrate is binding to the active site. But then in case of the competitive inhibitor, it is going and binding to the active site only. Okay, remember this way. This is the active site of the enzyme. Here the substrate binds, and the competitive inhibitor, it looks like this. It is very similar looking to the substrate. It is also coming and binding here. But then your non-competitive inhibitor, it is very small. It is coming and binding over here. This is an allosteric site. Okay, right. Now we will look at the summary of enzyme inhibition models. Now this is something that we have in the next slide as a question. So see, in competitive inhibition, what is happening? The competitive inhibitor is coming and binding to the enzyme and forming the enzyme inhibitor complex. It is preventing the substrate to come and bind. Okay. And it is, in competitive inhibition, the inhibitor binds only to free enzyme and not to the enzyme substrate complex. Okay. In uncompetitive inhibitor, it only binds to the enzyme substrate complex and not the free enzyme. And it forms the enzyme substrate inhibitor complex. In non-competitive inhibitor, it can either bind to the free enzyme or it can bind to the enzyme substrate complex, ultimately forming the enzyme substrate inhibitor complex. Okay. In mixed inhibition, what is happening? Competitive active site. Yes, correct. Competitive inhibitor is binding at the active site. Right. So now what happens is, in mixed inhibition, this involves inhibitor binding to both free enzyme and the enzyme substrate complex with different binding constants. So we will study about this dissociation constant for inhibitors as well very soon, and non-competitive allosteric enzyme. It is binding at the allosteric site. Correct. Similarly, for uncompetitive also it is binding at the allosteric site. Very good. Yes. Achha, now non-competitive, it can either bind to free enzyme or it can bind to the ES complex as well. Remember that. So this figure, the figures before and this one should give you a very good understanding of the different types of inhibitor models. Okay. Right. Now
Ma'am, hello.
Yes, yes.
Ma'am, can you please repeat the mixed?
Mixed inhibition. See, in mixed inhibition, what is happening? In mixed inhibition, this inhibitor can bind to the free enzyme. It can also bind to the enzyme substrate complex, but then the effects of its binding are different in both the cases. Okay, and we will study about that very soon. So see, one is Ki and the other one is alpha Ki. So alpha is actually changed Ki, which is the apparent Ki. So we will learn about that as well. So it is having different effects when it is binding at two different like states of the enzyme. Okay. But then in case of your non-competitive, see, it is having exactly similar effect, whether it is binding to free enzyme or it is binding to the enzyme substrate complex. It's all given here in this figure. Okay. Now we'll move on to plots in enzyme kinetics. So since Vmax is achieved at an infinite substrate concentration. So see, even if we extrapolate this, we are never going to reach the exact Vmax. And therefore, it is impossible to estimate the Vmax and hence Km from a hyperbolic plot. This is the normal Michaelis-Menten plot, which is hyperbolic in nature. Because of this difficulty, Michaelis-Menten equation is a so Michaelis-Menten equation was transformed into an equation for a straight line by Lineweaver and Burke. So they gave a plot which is a double reciprocal plot. Why? Because your reaction rate, that is 1 by V, that is V, and the substrate concentration, that is S, both are taken as reciprocal. So 1 by V and 1 by S, and that is plotted, and the slope gives us Km by Vmax. Okay, and we get the exact coordinate, that is 1 by Vmax, and exact coordinate 1 by Km, minus 1 by Km. These we are getting. Okay, so this is the Lineweaver-Burke's plot. So what happens in case of the different kinds of inhibition models when it comes to this Michaelis-Menten plot? So if you look at the Michaelis-Menten plot, you will see that the different inhibition models can be represented in this way. So in one, you know Km increases. In non-competitive, you know that Vmax decreases. This is how you can actually plot them using Michaelis-Menten curve. But then when it comes to Lineweaver-Burke plot, it is all the more visible and all the more comprehensible. So here we can see that in case of competitive inhibition, Km is increased. So this is how we see that the Km is shifting and Vmax is unaffected. See, it is coinciding at exactly the same spot. And the slope changes and the y-intercept remains the same. This is the y-intercept, which is 1 by Vmax. And the slope is changing. It is actually increasing. Right. In case of uncompetitive inhibitor, both Km and Vmax are reduced. And therefore, this line exactly shifts this way. So this also decreases. 1 by Vmax increases. So your Vmax gets reduced, and similarly for Km, minus 1 by Km goes towards the negative axis, and so Km gets reduced. Now we come to non-competitive inhibitor or mixed inhibition. Here this is the graph. The Km is remaining constant but Vmax is reduced. This is how you can understand the different models of inhibitor using the Lineweaver-Burke plot very nicely. Next up is Eddy-Hofstee plot. In Eddy-Hofstee, this is also a kind of deviation from the Michaelis-Menten plot, and they have somehow derived this particular equation from the Michaelis-Menten plot, and this is plotted between the rate of the reaction and the rate of reaction per unit substrate concentration. Okay, and this is how the plot goes. So the slope tells you minus Km, and the different intercepts give you information about the Vmax and Vmax by Km. Okay, now we go to different equations. Now these equations are very important because for solving any of the numericals you need these equations. So we have 3 slides of these equations, and then we will repeat previous slide. Which one is it? Eddy-Hofstee. Which one do you want me to repeat? Eddy-Hofstee slide?
No, ma'am. Previous. Second previous.
Achha, before this. Okay. This one.
No, ma'am. Just before.
Just before this one.
Yes, ma'am.
Achha,
yes, ma'am.
Okay. Achha, see, this you know is the Michaelis-Menten equation, right? V is equal to Vmax into S, that is substrate concentration, divided by Km plus substrate concentration, right? Now what happens is, normally when there is no inhibition, this is your graph. See, there is no inhibition and we are taking some values. Km is taken as 10 and Vmax is taken as 20. So this is the 20 and Km, you know that this is the Km half of Vmax. So it corresponds to this 20. Okay, substrate concentration. And this is for free enzyme. Now what happens is, in case of competitive inhibition, you know that for competitive inhibition, Vmax is not going to change. But then Km is going to increase. So what happens? This shifts towards the right. The graph shifts towards the right. And so we go and we see that Km is now becoming 20. So initially your Km, if you go from here, it was 10 and now Km is becoming 20. This is the one. And your Vmax, of course, it is not changing. So it is also 20. So inhibitor competes for the active sites and prevents the substrate. You can unmute yourself. Okay. If you're having any other doubts, you'll mute, you can mute yourself now and you can unmute yourself later. So yeah, we'll go back to this plot. Now what happens is, in competitive inhibition, you know that the competitive inhibitor is competing with the substrate to bind to the enzyme's active site. Because of that, what happens is, it is shifting the Km towards the right. It is decreasing the affinity of the enzyme for its substrate. But what happens in case of non-competitive inhibition? The inhibitor is reducing enzyme's maximal velocity, plus it is also not binding to the active site. Since it is not binding to the active site, the substrate is free to bind, and so the Km is unchanged. So Km is still here at 10. Km is over here. If you extrapolate this, you will get Km over here, that is 10. But then Vmax is reduced and it is 10 now. Okay. Is that clear? Okay. Great. So it is clear.
Yes,
We'll continue. So this is all about the different models of enzyme inhibition that you can understand using Michaelis-Menten plot. But once again, if you see, it is relatively not very comprehensive compared to the Lineweaver-Burke plot. Because in Lineweaver's Burke plot, it is very well explained. And we can very well see that the Vmax is actually reducing and Km is also actually changing. So this is a better representation of the different inhibitor models. Okay, you can go through this. And then we have the Eddy-Hofstee plot, which also can come as a question They can ask you the different, they can maybe give you this plot and they can ask you whether this is Line-Weaversburg plot or Eddie-Hofstee plot or Michaelis-Menten plot, and in that case you will have to say. Okay, and they can also give you some kind of derivation and a particular formula, and they can ask you which of these truly represents the Line-Weaversburg plot and which of this truly represents, let's say, Eddie-Hofstee equation. So that you must remember. Okay, now we come to the different equations in competitive inhibition. So this is competitive inhibition. That means the Km is going to increase, and this is the changed Km. So here alpha is a factor that describes how the binding of an inhibitor affects the enzyme's interaction with its substrate. Okay, and alpha can be found out using this particular equation, that is I plus concentration of the inhibitor divided by the dissociation constant for the enzyme inhibitor complex. This is your Ki. Okay. So Vi also changes. So this was your Michaelis-Menten equation, but when a competitive inhibitor comes in, this is how it changes. So with Km, you will have to multiply this alpha factor and you get this equation ultimately. Okay, so how do we get this Ki? Ki is nothing but the equilibrium constant between enzyme and concentration of enzyme and concentration of the inhibitor divided by concentration of the enzyme inhibitor complex. Okay, so the inhibitor decreases the apparent Km, that is the Michaelis constant, but the maximal velocity remains unchanged. Okay, sorry, it increases the apparent Km, right? It is a competitive inhibitor, so it is going to increase the apparent Km. So this is how you get an idea about alpha and you get an idea about the Ki. Okay, now we'll move on to the next slide in which we talk about uncompetitive and non-competitive inhibition. So uncompetitive inhibition, the Ki can either be this, in which the inhibitor is directly binding to the free enzyme, or it can be the next equation in which the inhibitor is binding to the enzyme substrate complex and forming the enzyme substrate inhibitor complex. Okay, this is also true for the uncompetitive inhibitor. Now in pure non-competitive inhibition, Ki is equal to Ki apparent. So both Vmax decreases but Km remains unchanged. Right. So what happens is, even your Ki, you can see that in both the cases it is Ki and Ki apparent, and these two equations are different, and therefore you can see that finally your Vi is also going to be different in both the cases. Okay. Now we come to something interesting, and that is Ki versus Km. So now the value of Ki is the dissociation constant describing the binding affinity between the inhibitor and the enzyme, while Km is the Michaelis constant and it is used to describe the kinetics of an enzyme substrate binding. So Ki is a thermodynamic parameter reporting the true affinity an inhibitor has for binding an enzyme. In contrast, voice breaking. Okay. Okay. Okay, you guys give me 1 minute. I'll just see if I can fix the connection. Right, let's see if this works. Tell me if you can hear me clearly. Okay. Okay, great. All right. So what happens next is the Ki is a thermodynamic parameter reporting the true affinity for an inhibitor for the enzyme. In contrast, Km is a kinetic parameter which gives the substrate concentration at which half of the maximal velocity is attained in the reaction. So Km is determined not only by the substrate's binding affinity but also how quickly the enzyme substrate complex is turned over into the product. In a rapid equilibrium where the substrate binding is fast binding comparing to the rate at which the enzyme substrate complex turns into product, Km and Ki are identical. Whenever this assumption is not met, Km will be larger than Ki. So this is something very interesting and you can have a look at this. So for this topic enzymes, I will recommend you to please go through either Voigt and Voigt's biochemistry or Leninger's principles of biochemistry. Okay, those two books are really the best for enzymes and most of the questions also are taken from either of these two books. So now we come to something very interesting and that is your inhibitory concentration. So what is IC50? There is lethal concentration LD50 or lethal dose 50 and there is IC50. Now these are things that are studied for drugs and poison or toxin. So I have attached the link to this website and this inhibitory concentration 50 is the concentration required to reduce the enzyme activity by 50%. Okay, and the concentration of the inhibitor required to do so is called the Ki. Just like your Km which was substrate, this is Ki. So 50% of the inhibition is attained at Ki concentration of the inhibitor. Okay, right. Now you try to answer this question. Question on enzyme inhibition in a mixed inhibition. The enzyme inhibitor complex EI has dash affinity for dash compared to dash. There are too many dashes, but then try to figure this out. So EI is the enzyme inhibitor complex. So enzyme inhibitor complex has higher or lower affinity to the dash. Compared to dash. Okay. You're saying B and I am also getting D as one of the answers. Okay. Anybody else who wants to answer? Try. EI is the enzyme inhibitor complex and this is mixed inhibition. So you know that in mixed inhibition, okay, I am getting D again as another answer. Let's see. Okay, so the right answer is C because EI, you know that it is going to bind to the substrate to form the enzyme substrate inhibitor complex. So the second dash should definitely be substrate, right? And this is going to have definitely lower affinity for the substrate compared to that of the free enzyme. That is why the inhibitor is there. That is its job. Correct? Now question on enzyme inhibitor and this is a competitive inhibition. So in competitive inhibition, an inhibitor binds to an enzyme at the other site compared to the active site only to the substrate reversibly at the active site covalently to the enzyme substrate complex. Tell me. Yes, very good. So you guys have actually understood this. Very good. Yes, you all are correct. It is C. Competitive inhibitor is always going to bind reversibly at the active site. So now what another interesting thing is that, let's say there is a competitive inhibition going on and you suddenly increase the substrate concentration hugely. Then what happens is the substrate goes and competes with the competitive inhibitor, and more of the substrate can now bind to the active site, and the reaction which had initially slowed down, this actually proceeds properly and it can be recovered. So remember, this is always reversible. Okay. This is another question. This is how you might get questions. So there might be equations and they might ask you whether which of the equations properly represent the Lineweaver-Burke plot, which of the equations actually represent Eddy-Hopstein plot, this way. Okay, so you have to remember in which plot what are the different axes, and so from there you can derive using the Michaelis-Menten equation exactly what should be the equation for a specific plot. Okay, so see, they are asking you which of the following equations represent the Lineweaver-Burke plot properly. So all of these are, you have to first convert them into y is equal to fx plus c format, because this is a straight line, and so you have to convert it into the straight line equation and then find out. Okay, so you are saying that it is B. B is 2 and 4. Right. Okay, let's see. The right answer is B. You can solve these and you can see for yourself. Okay. These are the kind of questions that you can actually expect in your exams. These questions will come, and for these you need to know the axis for different kinds of plots and the Michaelis-Menten equation and how to derive each. So now this is another question. If you remember the Michaelis-Menten plot for the different inhibitor models, you will be able to answer this. So the graph below represents reaction rate versus substrate concentration plots for an uninhibited enzyme, which is orange, and the same enzyme in the presence of two inhibitors. One is A and the other one is B. Acha. So I am already getting answers. Okay. A is competitive and B is non-competitive inhibitor. Think properly and then answer. Yes. So you guys have actually understood the class. I am glad that you understood. Let's see. It is D. See, this has to be competitive inhibitor because we see that Km is increasing, right? But Bmax is unchanged. But then in case of B, we see that the Km is rather unchanged and Vmax is decreasing. So that is the thing for non-competitive inhibitor. But then in case of uncompetitive inhibitor, what would happen? Both of these would decrease. Both Vmax and Km was supposed to decrease when it comes to uncompetitive inhibitor. Both of these reduce, right? So yes, you are correct. This is definitely non-competitive inhibitor. So we'll go to the next one. Acha, try answering this. What kind of inhibition is depicted in the diagram shown below? So you have to see whether the inhibitor is binding to the substrate's binding site, whether it is binding to a different binding site, whether it is binding to free enzyme, whether it is binding to the enzyme substrate complex, and whether it is binding to both. That is what you need to see. Mixed inhibition. Okay, why is that so? C is non-competitive inhibition. Let's see. Okay. One of you is saying D. The other one is saying C. Let's see. It cannot be competitive because you know the right answer, right? It is not binding at the substrate's binding site. So it is definitely not competitive. Okay, let's look at the right answer. It is non-competitive inhibition. Why? Because in non-competitive inhibition, the inhibitor binds to free enzyme and it also binds to the enzyme substrate complex. And it finally forms the enzyme substrate inhibitor complex. So that is the property of non-competitive inhibition. Okay. Right. Okay. We'll go to another very interesting question and we'll try to solve this. In the scheme, how it differs from mixed inhibition? Right, that is a very good question. How does non-competitive inhibitor differs from, sorry, one second. Can you see the presentation? Okay. How is it differing from mixed inhibition? Okay, can you guys see? Not sure. Yeah, can you see the presentation? So the question, okay, great. Yeah, okay. So the question is, how is non-competitive inhibitor different from mixed inhibition? So mixed inhibition and non-competitive inhibition, these are similar. Okay, non-competitive inhibition is a special case of mixed inhibition. So the only difference is that in case of non-competitive inhibition, the substrate binding has no effect on the inhibitor binding. So you can see that Ki, it is Ki in both the cases. Whether the inhibitor is binding to free enzyme or it is binding to the substrate, it is going to remain as Ki. But then in case of mixed inhibition, there is a change in the inhibitor's dissociation constant when it actually binds to the enzyme substrate complex. So in mixed inhibition, there is involvement of inhibitor binding to both free enzyme and enzyme substrate complex with different binding constants. That is the difference. We'll go back to the question. Now, since in this slide they have not shown the Ki, so it would be difficult for you to say whether this is mixed inhibition or non-competitive inhibition, because you cannot exactly see how the like dissociation constant for inhibition, inhibition constant is changing or not. Okay, so you can read more about this, why it is non-competitive inhibition and why not mixed inhibition, and you can tell me about it in the next class. Okay, easy in graph but in picture. Yeah, exactly. You have to read about this more and then we can discuss in the next class. Okay, right. So now we come to a relatively tougher question. In this question, they are asking you what is going to be this 1, 2, 3 and 4. What does 1, 2, 3 and 4 going to represent? Okay, try to see. Now for the fourth option, you have beta into Kp for all. For the first, second and third, what do you think are going to be the representations for 1, 2 and 3? D. Okay. Any other answer? It is D. Okay. Do we have any other answer? C. So I'm getting different answers. Okay. C. The right answer is C. Ki, we studied in those formulas. It is there in the slide. Ki is your E into I divided by EI complex. So it has to be this one. 1 would be Ki. In case of alpha, you know that ESI was involved, and so your 2 is ESI and ES, right, plus I. So this would be alpha into Ki. Now we come to this one, that is the third equation. So this is alpha into Ks. Alpha Ks, this is ESI and EI along with S. So always remember this. When inhibitor is involved, it is becoming alpha Ki. And when the substrate is involved, it is becoming alpha Ks. And this beta into Kp is the rate constant for the breakdown of ESI to P. When it was ES to P, it was Kp, but then a term beta is getting introduced when this is enzyme substrate inhibitor complex, and that is breaking down into your product and enzyme inhibitor complex. Okay. So that is the right answer. C is the right answer for this question. Remember all of these formulae. We have discussed about these formulae in this particular slide. Yeah, this one. So see, Ki, remember it is this. So when Ei and E and I are there, that gives you Ki. What is alpha into Ki? Then your ESI is involved. ESI and ES and I, that is involved. So that is how you can distinguish. Okay. Now we go to another very interesting topic, that is what happens if there are more than one substrates in a reaction. It is a multi-step reaction. So there can be different kinds of multi-steps reactions. And you know that for these multi-steps reactions, the Michaelis-Menten equation holds no good. Okay. So there are 3 different types of multi-state reactions, multi-substrate reactions that we are going to learn about today. They are ordered sequential reaction. Another one is random sequential reaction. And the third one is ping pong by double displacement reaction. Okay. So we'll have a look at all of these. So what happens in ordered sequential? So initially you see substrate A is binding, and then substrate B is binding, and then the product is getting formed and it is leaving. In case of random sequential, we see that it is not necessary that A will bind first or B will bind first. Anyone can bind first and then we are getting the products. But then in ping pong by double displacement reaction, when A is binding, it is producing the product, and then B is binding, and then there is kind of a displacement. Okay. So we will learn about them in details. Now see the reactions. These are actually by reaction. By means by substrate reactions. So these reactions fall into 2 groups. One is sequential, one is ping pong double displacement. So these names indicate whether the reactants must bind to the enzyme at the same time for the reaction to occur, that is sequential, or if a product must leave before the second reactant can bind, which is ping pong. So just now we studied in the previous slide that in ping pong reaction the product had to leave before the next substrate came and bound, right? So sequential reaction, in these reactions both the reactants must be on the enzyme for the reaction to happen. Both the reactants must be on the enzyme, bound to the enzyme. If this is a group transfer, the implication is that the group is transferred directly between the two reactants sitting on the enzyme's active site. So these reactions are sub-classified as ordered or random. So sequential reactions, you know, there can be ordered reactions. So in ordered reaction, the enzyme bind in different order with the substrate. If the mechanism is ordered, the substrate will add to the enzyme as A first, then B, and then C, and finally give rise to the products. So one of the examples is NAD plus dependent dehydrogenases. Okay, now we come to random sequential bye-bye reaction. So the order of binding is not important, indicating that the binding sites for each substrate and the product are fundamentally independent of each other. So in sequential mechanism, usually no intermediates are formed. Okay, but then in case of your ping pong bye-bye mechanism, see, initially A is coming and the product has to leave before B comes and binds, and then again another product is leaving. So in the second major class of bye-bye reactions, one of the products is released before the second can bind, and a stable enzyme intermediate is occurring in these reactions that reacts with the second substrate. So this reaction intermediate is the one that is interacting with the second substrate. Often the enzyme is transiently covalently modified in order to transfer a functional group from one substrate to another. The substrates are not in contact with each other on the enzyme. This is the major difference between the ping pong bye-bye and the sequential ordered reactions, sequential reactions, right? There is no interaction between the substrate, rather the chemical group is getting transferred from the substrate to the enzyme and forming an intermediate that is interacting with the next substrate and forming the product. So this type of mechanism is seen in serine proteases and common in aminotransferases and some flavoenzymes. Okay, so uridyl transferases. So any kind of transferase, you know that it is going to take a particular chemical entity from something and put it on to something. So it is going to carry that chemical entity in the process. So that definitely transferase would be involved in ping pong bye-bye mechanism. Uridyl transferases, and they provide interesting structural examples on how different substrates share the same active site. So ping pong mechanism has intermediates. Okay, right. So these are the different graphs that you can see for ping pong bye-bye interactions, and this is the graph for sequential interaction. You can read more about this if you're interested. But for our MCQs, I think this much knowledge is sufficient. So we'll go on to the question. Right, this is again a question, and different your equations are given, different chemical reactions, and from here you have to identify which is a random ordered interaction, whether it is sequential ordered interaction, and whether it is ping pong bye-bye interaction. So if you have understood until now, it should be easier for you to answer. Okay, hello everyone, I'm back again. I had lost the connection, and so we'll continue with this. If you have answered any of the questions, please answer once again in the chat box, because I have missed out. So please answer once again.
आवाज नहीं आ रही मैम.
vanished. I know. My connection was lost. Oh, achha. Can you hear me now? Okay, great. Can you please answer this question once again in the chat box? My connection was lost and I had to leave the chat. Please answer once again in the chat box. Full screen mode. Please answer. Okay, so you guys are saying that it is D. The first one is why do you think it is D? Let's see. Let's see which is the right answer. Very good. It is D. So in ping pong reaction, double displacement pathway is followed. So initially you are getting this intermediate. E star is the intermediate, right? And then there is always kind of displacement and that product is P1 is going out and only then your S2 is coming and binding and it is giving the product P2. So this is definitely double displacement reaction and this is your ping pong reaction. Now we come to this one. It is very clear from here that this has to be your random sequential interaction, right? And this one is your ordered sequential interaction that is happening. So at the same time, S1 and S2 are bound on the same enzyme and we are getting two different products, right? So if whatever I have told you, and voice is not audible once again. Okay, can you hear me? Acha. So based on whatever we learned about these different kinds of reactions, I hope you will be able to answer the MCQs. Okay. If you're interested in these, you can learn more about these from all the links that I have provided, website links, and from the books. Okay. Now, for answering one more question. Now, this question is taken from Voigt and Voigt's biochemistry. Okay, so once you start reading, you will see that you will read about different kinds of drugs that are used to treat HIV and how these drugs work. Here in this slide, this is taken from a review paper and we see that there are different kinds of inhibitors. There is protease inhibitor. Protease inhibitor example is ritonavir, and there is nucleotide analog drug which is remdesivir. So any enzyme that is going to act on viruses, most of the times you will see that they are having this VIR last name, and if it is made from some kind of antibody, monoclonal antibody or not, and all, then you will see the last name ends with MAB. Okay, so and that is how the naming of these drugs go. And nucleotide analog means that it is some kind of modified nucleotide. It is looking like the nucleotide structurally, but then it is having some kind of chemical group attached, and that is going to prevent the, let's say, transcription and replication of the viral RNA. So this is how different kinds of drugs are used. So now this is one question. Okay. So for this question, you have to study Voigt biochemistry. Now you can try to answer this question. So we read about ritonavir here. Based on that, try to answer this question. Dash inhibits the HIV protease enzyme through A or an dash pathway, forming an enzyme inhibitor complex. So if enzyme inhibitor complex is formed, then what kind of pathway should it be that prevents the cleavage of specific polyproteins Y inhibits acetylcholinesterase through dash pathway by reacting with serine at the active site. Okay, so you are saying that it is A. Okay. The others try to answer too. Okay, A and B. These are the two that I'm getting. Okay. Yeah, it will either be A or be B, because we saw here that this was a protease inhibitor. Right? Okay. So the right answer is B. Ritonavir is a reversible inhibitor and it inhibits the HIV protease enzyme by forming an enzyme inhibitor complex. Enzyme inhibitor complex. Okay. And prevents the cleavage of the specific polyproteins. And diisopropyl fluorophosphate or nerve gas is an irreversible inhibitor that reacts with serine at the active site of the acetylcholine esterase to form a covalent bond. Okay. So, read about this in Voigt's biochemistry. Okay. Right. Now we come to one more question, and for a clue, this figure is given to you. This is the active site of carbonic anhydrase. Now, based on whatever information is given on the slide, try to tell me which is not correct for carbonic anhydrase. And only one is the right answer. So, you can try to answer based on whatever is said. Okay, the question plus the clue. So, option A says that it can increase the rate of conversions of carbon dioxide to carbonic acid and bicarbonate ions up to 1,000,000 folds. It presents in blood cells. It is present in blood cells. D is malfunction of this enzyme can lead to several diseases like loss of acid production in the stomach, kidney failure, etc. Which is false. They are asking you which of these is not true. Look at the clue properly. Very simple. Clue is also given. Try to answer. So option number C is telling you in its active site, zinc is coordinated with 4 histidine residues. Yes, right. It is C. C is not true. Exactly. Why is C not true? Because we can see that there are 3 histidine residues that are binding with zinc. Another histidine residue is not binding. The other residue that is binding is what? Yes, the right answer is C. Okay. Zinc is coordinated with 3 histidine residues and not 4. Okay. Now we come to isoenzymes and alloenzymes. Now this is again a topic. It's in insulin. If I'm not understanding. Excuse me. Can you please explain? I did not understand. Okay, Ajay, you can explain yourself if you want to say anything to us. Okay, I did not understand what you've written in the chat box. So you can explain if you want. Zinc plus 4-histidine present along with Cu in insulin hormone. Okay, is that so? Okay, thank you for the information. Okay, we'll try to remember this. So we'll carry on with isoenzyme and alloenzyme. Right. So this is also a place from which you get questions. What are isoenzymes or isozymes? Now see, there can be different kinds of events like gene splicing. There can be gene duplications, and because of this, from the same gene that is undergoing duplication to give similar gene, or there is some kind of alternative splicing and it is giving rise to another gene with maybe slightly different sequence. This gives you proteins with very slight modification in the sequence, because of which different physical and chemical properties can be acquired by these proteins, although they are probably similar in function. Okay, so isozymes or isoenzymes are different variants of the same enzyme, but they have identical functions and are found in the same person. This definition includes enzyme variants produced by different genes. You can see gene B, C, D and E. All of these are variants of the same gene that are now different, and they are producing different isozymes of the same enzyme. So, they are produced by different genes and thus representing different loci, referred to as isozymes, as well as enzymes produced by different alleles of the same gene. They are described as allozymes. So different alleles means, for example, for your blood group, you know that there are two different alleles of the same group, or two different alleles give rise to a blood group in a person, right? For example, if your blood group AB, then it is allele IA and IB. So allele means the two different variants of the same gene present in the two chromosomes, diploid chromosomes, right? So that is your allele. So here, if two different alleles are producing two different enzymes, these are called allozymes. But if they are produced by the exact same gene from the exact same locus, then it is called it is produced by different genes, then it is called isozymes. Okay. So isozymes arise from gene duplication and alternative splicing events of the single gene. Allozymes are variants of an enzyme found in different individuals. So, like blood group, it is different in different individuals, right? So, the same individual cannot have three alleles present at the same time. That is not possible. Single individual will not have I and IA and IB at the same time. That is not going to be possible. And similarly, your allozymes, they are present across the population. So they are variants of the same enzyme found in different individuals within the same species, differing due to genetic variation arising from different alleles of a gene within a population. Because of the presence of these isozymes, it is very difficult to delete some gene from the human genome. Okay, let's say there is some kind of enzyme. For example, this allele, allozyme, same gene. Iso is different gene. Yes, that is right. Allele, same gene, but then different allele is giving rise to your allozyme. But same gene that is undergoing duplications and alternative splicing to give rise to different genes, that is giving rise to isozymes, which are functionally very similar but this property wise different. Okay, so there can be isozymes of the same enzyme existing in different regions of your body. For example, there can be pyruvate kinase existing in the muscle and in the liver, Both of these are functioning only slightly differently. Okay, this is how you can remember. So see, in this diagram you can see that this isozyme enzyme, it is maybe present in cancer cells. Okay, and then how do we get rid of this? Because if we delete this, then some other variant is there in the body, and that is why it becomes very difficult to delete genes in the human body, because there are so many variants of so many different genes in our body. And that is why to study the effects of a particular gene, we take eukaryotic species with which are less complex compared to us. For example, mice, right? Okay, I've talked a lot about this. Now you try to answer this question on isozymes. Try to answer. Which of the following is not true for an isozyme? They differ in the amino acid sequence but catalyze the same chemical reaction. They have the same Km values. They vary in chemical and physical properties, which is not true. And they are differently regulated. A is not true, you're saying, which is not true. Try to answer. Try to recall the example that I gave you. B. They have same Km values. Okay. Let's look at the right answer. Okay. So if you look at this question, isozymes, they are coming from different genes which are having very similar sequences. Why? Because there is gene duplication or alternative splicing. Right? So the genes are having similar sequences. That means they are going to code for the enzymes with very similar sequences with slight difference in the amino acid sequence, because of which they will differ in amino acid sequence. As a result, they will have different physical and chemical properties, but they will catalyze the same reaction. Pyruvate kinase in muscle and pyruvate kinase in liver, both are going to catalyze same reaction, giving rise to pyruvate, right? Phosphoenol pyruvate to pyruvate. But they are going to have different Km values. They are going to act on the substrates differently. That is the thing. Let's look at the right answer. They have different Km values and they have different affinities for their respective substrates. And they are also regulated differently. That is something that you have to remember. We will look at the next slide how this happens. Okay. Right. So this is another question. You try to answer this, and very soon we will look at pyruvate kinase and how it is differently regulated under different environmental conditions. So this is a question on enzyme. When a product accumulates, okay, so you are saying that it is feedback inhibition. So when a product accumulates in a cell beyond an optimal amount, its production is decreased by inhibition of an enzyme involved in its synthesis. After the product has been utilized or broken down and its concentration is decreased, the inhibition is relaxed and the formation of the product resumes. This inhibition is a mechanism by which the concentration of certain cells constituents is limited. What type of inhibition is this? Yes, very good. This is feedback inhibition or product inhibition. Okay. So see, in feedback inhibition, the enzyme activity is suppressed as it participates in a sequence of reactions by which a substrate is synthesized. Substance is synthesized by a product of that sequence. And this inhibition mechanism limits the concentration of certain cell constituents. Okay. And this is your pyruvate kinase in the liver and in the other tissues, for example, muscle. So see, it is regulated by the concentration of glucagon, and it is actually acting on the same substrates, but then it is regulated by different kind of entities. M and L isotypes refer to the specific forms of the enzyme in muscle and liver respectively. Okay, so there is ATP, there is acetyl coenzyme A, there is long chain fatty acids. All of these are regulating the activity of pyruvate kinase. Similarly, only in the liver, glucagon is affecting the activity of the pyruvate kinase enzyme. Okay, here in this slide, we get to see this even better. So see what is happening in M type. There is M type means that is in muscle. There is allosteric regulation. So AMP, fructose 1,6 bisphosphate are activating pyruvate kinase. ATP concentration, acetyl coenzyme A and alanine, these are inhibiting pyruvate kinase concentration. Okay, in the L type, that is liver, insulin and glucagon are influencing pyruvate kinase activity, but then the reaction is same, pep to pyruvate. This is also pep to pyruvate, but then the other entities influencing it are different. Okay, besides that, if pyruvate kinase deficiency is seen, then there can be hemolytic anemia, red blood cells swell and they lyse, they are breaking up. So RBCs lack mitochondria and rely on glycolysis. So when there is pyruvate kinase deficiency, then what happens? The cell, there is no ATP for the sodium potassium pump to maintain the cell shape, and they change in shape and then they lyse. So this is something that you have to keep in mind. Now we will go to another enzyme, which is acetylcholine esterase. So acetylcholine esterase is an enzyme which is acting on acetylcholine and it is forming acetate and choline molecules. Now this choline is again going and acetyl coenzyme A is coming and then together gives rise to acetylcholine, and this is involved in the synaptic vesicle signal transmission. Right. So this is a synaptic neuron and this is a postsynaptic neuron, and between them there is release of acetyl coenzyme A and then signal transmission. Okay. So this is all about your acetylcholine esterase, acetylcholine esterase. Now we go to RNase A. RNase A, if you remember, this is an enzyme which is going to act on RNA and it is going to chop off RNA. So this acts on single stranded RNA and it is going to cleave RNA. In the active site, this enzyme has histidine and lysine molecule, lysine residues. So this is ubiquitously present. RNase A is present on your hand. It is present maybe on your computer. It is present on your desk. And because of the presence of RNases and because of their very high stability, it is very difficult to work with RNA in the laboratory, because if you're not careful with your RNA samples, RNase A will be there everywhere and it will just cleave off the RNA. Okay, so even when you autoclave it, autoclave means heating at 121 degrees centigrade, RNase A is more studied. Yes, RNase A is very well studied, and RNase A is something that is highly stable and very resistant to even heat denaturation. So if you autoclave the water once, it is not going to kill RNase A. RNase A will still be persistent, and that is why you need to autoclave your water for any RNA based experiments minimum twice. Okay, now we go to a simple question, and I think this is the last question for today. You try to answer this. I have told you so many stories about pyruvate kinase, about acetylcholine esterase and about RNase A. Now you try to answer this question. Yes, you're right. At neuromuscular junction or synapse, acetylcholine is released. Yes. Acetylcholinesterase hydrolyzes. Which of these is true? So acetylcholinesterase hydrolyzes the neurotransmitter acetylcholine in the synapse and terminates its activity, and it acts as a non-competitive. Which of these is true? Acha, you're saying that it is 1, 2 and 3, all of these are true. Okay. And then you're saying that all of these are true, all the 4 are true. Yes. So all those who are saying that A is true have seen the picture of RNase A properly. Okay. Yes. A is the right answer. That is because RNase A has 2 histidine residues and only 1 lysine residue in its active site, and not 2 lysine and 1 histidine. So if we go back to this figure, you can see that there are 2 histidine residues and 1 lysine, whereas here it is given that there are 2 lysine residues and 1 histidine. Besides that, we know that both M and L type of isozymes, pyruvate kinase, are allosterically regulated. That is there in one of the slides just previous to this one. Plus, pyruvate kinase deficiency leads to hemolytic anemia. This also we have learned just now. And acetylcholinesterase is acting on acetylcholine, and this is preventing the activity. That is also something that we know. Okay. These are the different references or the different websites and books and research articles that you can go through. So after that, we are done with the lesson today. Thank you so much. If you have any questions, you can ask me. Otherwise, we can end the session. Thank you for your patience. Thank you for your cooperation. We'll meet again in the next class on next Tuesday. Thank you so much. The lecture material of week 1, 2, 4 and 5 are still not available for download, and the problem has not been fixed. When I click on the lecture material of the course outline panel for a week and then visit the hyperlink provided, either I get empty G drive folder. Sorry, the file you have requested does not. Acha. Oh, is that so? Because I have uploaded all of these PPTs. Okay, all are available. You can refresh once, okay? You can refresh and you can check. Because I have uploaded all of these. So all of these should be available to you. Even if you are seeing that there is some kind of issue, you can mail NPTEL saying that this is the issue. I am not able to access the PPTs. I hope they'll. Okay, so I'll basically give you my mail ID, right?
एक minute. Hello, ma'am. If any doubt regarding weekly session, I will just ask you. So I will need your email ID, either any contact.
Yes, sure. See, I have given my email ID to you in this chat box. So you can take a screenshot, you can copy this, and if you have any issues, you can mail me, or in the next class, you can ask me. Okay, whatever is comfortable for you, you can do that.
Okay, thank you, ma'am.
Thank you so much. Okay, if you guys do not have any other questions, any other suggestions or feedback, then we can end the session today. Thank you so much. Thank you so much.